Hello everyone and welcome to the L Microbiologist channel. Today's video will be the first episode of a medical bacteriology course about Staphylococcus aureus. We will be discussing its morphology, its general characteristics, its behavior and virulence factors, the diseases it causes, and how to choose the best antibiotics. From the name Staphylococcus aureus, we can conclude its shape. Coccus means round and staphylo means grape-like. So Staphorious appears as round colonies in grape-like clusters which can have a golden aura as a result of an exopigment that it produces. This golden aura is what gives Staphorious the aureus part of its name. Staphorious bacteria can easily be identified using the gram stain method, where it appears violet in colour, indicating that it's a gram-positive organism. Staphorious can be found as part of the normal flora on the skin, the mucous membranes, and in the upper respiratory tract, where it helps to prevent infections from other pathogenic organisms. But under different environmental conditions, such as a change in humidity, pH, sweating, or if natural barriers like the skin are damaged, it can become an opportunistic pathogen. The bacteria can cause a variety of different superficial infections, such as impetigo, boils, carbuncle, or abscesses, and may also cause more serious illness like sinusitis or osteomyelitis if the infection becomes deeper seated. Staphorious can also invade the bloodstream in its vessels, causing infections like septicemia, or in other different areas of the body as it gets transported around. These can include the brain, where it causes meningitis, the heart, causing endocarditis, the lungs, where it causes pneumonia, or the kidneys, leading to urinary tract infections. You will find that most Staphylococcus infections are superficial, which is why over-the-counter creams, ointments, and nasal solutions from the pharmaceutical or cosmetic industries can be used to effectively treat infections. Staphorious is considered one of the most pathogenic opportunistic bacteria due to its specific virulence factors, which includes the production of a range of different enzymes. One example of this is Coagulase, which binds to and activates prothrombin, converting soluble fibrinogen into fibrin, which aids in blood clotting. The activation of the clotting process also evokes an adaptive immune response in infected individuals. The production of Coagulase is very specific to Staph aureus and can be used to differentiate it from other species in the Staphylococci genus. After the cells are exhausted by Staph aureus, it produces staphylokinase, which is an enzyme that disrupts the clots formed by Coagulase by activating plasminogen to form plasmin. Staph aureus is disseminated along with the clot, allowing it to move to infect other cells. Hyaluronidase is another enzyme produced by Staph aureus, which breaks down hyaluronic acid, which decays the connective tissues and skin and helps to spread the bacteria. Among the enzymes that increase its virulence and enable it to evade the immune system to cause infections is lipase. Sebaceous glands in the skin secrete oils, especially during sweating, that can be utilized by Staph aureus as a source of nutrients to help it cause local infections. Staphylococcus aureus can also produce thermonuclease, which is a heat-stable nuclease that breaks down DNA, leading to cell damage and death. The thermonucleases produced by Staph aureus can be used as genetic markers, which are detectable in food samples. The second type of virulence factor is the production of toxins. Staph aureus can produce toxic shock syndrome toxin, which binds to antigen-presenting cells which are supposed to recognize foreign microorganisms and eliminate them, leaving behind an immunological memory against future infections. But when Staph aureus produces the toxic shock syndrome toxin, it causes a huge release of cytokines that in turn cause a high fever, rash, hypertension, sudden shock, and death. Another toxin produced by Staph aureus is leukocidin, which targets and kills human granulocytes and monocytes. It can also produce hemolysin, which breaks down red blood cells and hemoglobin. Staph aureus can also release exfoliative toxins, which are serine proteases that separate the epidermis from the dermis, deepening skin infections. As well as skin infections, Staph aureus can also cause food poisoning through the release of enterotoxins, which can lead to vomiting, diarrhea, muscle spasms, and abdominal pain. Now we'll move on to the most important virulence factor, which is biofilm formation. Staphorious can universally attach itself to surfaces and produce extracellular polysaccharides, which produce a biofilm that isolates the bacteria from external environments, including antibiotics, which contributes to its resistance. Even if antibiotics can penetrate the biofilm, Staphorious may contain antibiotic resistance genes, which render them ineffective. Alexander Fleming discovered penicillin at the expense of Staphorious, although now many strains of Staphorious are resistant to penicillin due to the production of penicillinase enzymes, which inactivate penicillin's mode of action. Many strains are also resistant to methicillin and are known as MRSA, or vancomycin, or VRSA. Because Staphorious is resistant to many of the beta-lactam family of antibiotics like penicillin and methicillin, clavulanic acid is now added to many of these antibiotics to inactivate the beta-lactamase producing enzymes secreted by the bacteria to make them more effective against resistant strains. An example of this is amoxicillin with clavulanic acid, which is a good choice for potentially resistant strains of Staphorious. As well as this, other treatments such as vancomycin, ciprofloxacin, and tetracycline are all effective antibiotics. When it comes to identifying a Staphorious infection, the first step is often a gram stain, which, as we've already mentioned, appears as a purple gram-positive coccus which may appear clustered. The catalase test can also be used as a rapid method to differentiate between gram-positive Staphorious and gram-negative streptococci. To differentiate Staphorious from other Staphylococci group members, we perform a test to identify the presence of the coagulase enzyme. Because unlike any other member of the Staphylococci group, Staphorious is coagulase positive. The visible gold pigment we referred to earlier, which results from staphyloxanthin exopigments, can also be used to identify Staphorious when grown in different culture medias, like nutrient agar or tryptone soybean agar. Selective agars like mannitol salt agar may also be used to confirm the identification of Staphorious. On this media, Staphorious produce yellow colonies and survive a high salt percentage, around 7.5%. Its ability to tolerate a high salt environment is very characteristic of Staphorious because it is a known halophile. Baird-Parker agar is another selective media for the isolation and identification of Staphorious because it contains potassium tellurite and egg yolk. On this media, Staphorious produces black colonies with clear zones surrounding them. The black colonies are a result of the reduction of potassium tellurite into tellurium, while the clear zones are due to the ability of Staphorious to produce lethacinase and lipase enzymes that utilize egg yolk lipids. Blood agars are also important media in the identification of Staphorious because it has the ability to cause complete lysis to red blood cells and can produce beta hemolysis due to the production of hemolysin toxins. The final media example is toluidine blue DNA agar. Staphorious can produce thermonuclease enzymes that denature DNA present in the media, which produces pink colonies. And that's the end of this episode. Thank you so much for watching. I hope you find this video helpful.